Okay, let me show you now how you will continue if you want to be more, follow more standard procedure and add the biological description of your data set. And the way we typically do that, we start by creating our biosample. So I know the biosamples that I have, and I will try to enter the description for that. So I will navigate down to the biosample section, click Add Records, and go and enter my first biosample. So as you can see, the data set is already populated because we created this biosample from this data set. So we know it's already pre-populated. Then we have the next two fields is subject ID and local identifier. So subject ID, we use that when it's a human sample. For animal sample, we use the local identifier. And in many cases, we've encountered many groups that they have pretty well established schemes for labeling their samples. You can enter that here if you have that. If you're one of those groups, then you can use that identifier here. We would like you to do that. If not, you can enter something that makes sense within your experiment. For example, I will enter E14.5 mutant replicate 1, because this is my first sample of that type. I know that I performed the experiment in 3 identical samples, so I will, consider them like replicates. Species is going to be mouse. Specimen, I will select the term that best matches from what I did. In this case, let's say it's tissue specimen. Gene, again, LHX6. In many cases, you don't have to repeat this. I just did it before because I wanted to show you, but if you just do it at the biosample level, it will then automatically be populated at the higher level. Then I will specify the genotype. Again, stage, E14.5, anatomy, root of molar tooth, the term from Uberon. I think that's pretty much everything I have. Let's say I have the liter information from that mouse. Let's say I know when the sample was collected. Let's say I did it last week. And this is basically pretty much all the information that I need to describe one biosample. Let me show you something that, even though it looks like complicated, we have actually our interface is very good and very efficient to create and enter a large number of these biosamples of experiments. And we have people that have curated data set with thousands of them, hundreds of them in a very reasonable time. And one of those tricks is using this clone mechanism. So I know that I want to create 3 biosamples that are pretty much the same. So I will enter 2 here in the quantity of clones. I will click clone, and this basically replicates these entries, and I will just change the only things that I know when to change, because I have 3 replicates, all the other information is the same, so the only thing I'm going to change is the local identifier. So next I will click save, and then that tells me that I created these samples. I will switch back to my previous data set tab, and see now if I click empty sections, I have created these 3 biosamples. So the next step will be to create an experiment, telling what did I do on these samples. So I will create one experiment, and the idea, the kind of the rule that we try to follow is that we try to create one experiment for each set of identical biosamples or replicates. So if you will have three replicates, 3 mutant replicates, and 3 control replicates, they will create one experiment for the controls, one experiment for the mutants. If you were to have in this same data set, you are combining imaging with the sequencing data, then you will create one experiment for the imaging of the mutants, and then another experiment for, let's say, single cell experiment of the mutants. and you will group all those biosamples within that experiment. So right now I will create one experiment for the fluorescence microscopy of these three identical or replicate samples. So the first field is the experiment type. I will again search for fluorescence microscopy. I will give you the local identifier. Again, if your group has some scheme for that, you enter that in here. If not, you just give something that makes sense. Mutant. So that means there's a fluorescent microscopy of the mutants. All these other fields are for other type of experiments, not for microscopy experiments, but if you were to be using a DNA, like ChIP-seq or RNA sequencing or single cell, then you will add some of this additional information in here. So here we'll click Save. And at this point, I basically enter all the sort of structure of what I need to know. So I describe my biosamples, I enter my experiment, and now I'm ready to upload the data. And so at this point, the only thing you need to do is to tie the experiment to the biosamples and then upload the data to each of these combinations. And the way we do that is by defining these elements that we call replicates. So for each to connect the biosample to the experiment, we generate the replicate, and then we will add that data to a replicate. So I dive down into the experiment that I just defined. I go to the replicate section, I click Add Records, and here I start selecting. So in the biosample field, I see that all the biosamples for these data sets are shown in there. So I will pick the first one. That will define my first replicate. Since I need to do 3, again, I will use this clone feature of our interface, and I will change the data, the biosample, and change the replicate number so that, they look ordered. So now I have defined the 3 replicates, and now if I go to my experiment, I see the 3 replicates that link to that biosample. And if I navigate back to the data set, so I can navigate back to my data set using 2 ways. One is switching to this other tab that I've been doing, but sometimes it's useful to just follow the link in here. So I can click here, and now if I hide my empty sections, I see what I've done, and I can see that now the experiment has all the information coming from the 3 biosamples that I selected. I basically link into this.